is that people don't understand what vaccines are, what they do, how they work, and it's a lack of information and a lack of the facts. And I think that's what is essentially one of the things that's dividing the country these days is just I won't say ignorance of the fact, because the public, I think part of the problem has been the medical profession itself, or the research profession is the facts haven't been communicated properly to the general public. And so if you don't have the facts, how in the world can you make rational decisions? And I think that's the basic problem. So the idea behind this talk today is to get this to work. Just the facts, okay? So we've all seen Dragnet, Sergeant Friday, ma'am, just the facts. And so that's what I want to do today, is give you the facts. My bias is probably going to show through, so like I said right up front, what my bias is, but I'm really trying to suppress that. And what I want to do is essentially educate everybody here to what's going on. Okay? That's the idea. Okay, so the talk was the good, the bad, and the ugly. Let me start off with the ugly. And this is a reminder of what the world looks like in the absence of vaccines. Smallpox. Smallpox likely infected humans for at least 10,000 years. Death rate, 30%. 80% children. Estimated just in the last first three quarters of the 20th century, 300 to 500 million deaths. That's smallpox. People in this room have never experienced smallpox. I haven't, because we've been lucky. Influenza, 1918 epidemic, 17 to 100 million dead over the world, 1 to 5.4% of the global population. The reason that the estimate is 17, it's such a wide range, is because during that time period was World War I, and governments all over the world kept the information secret. So a lot of the deaths went unreported. What you see in this picture is morning call in New York City. Every morning, these hearses or ambulances would show up to people's houses and they'd remove the dead. People didn't die mostly in hospitals then, they died at home. So and this was typical of all these cities in the United States. So this is morning call in New York City, and they're loading corpses to take them away. Polio. Now, people in this room may have some experience in polio. 1952 epidemic, 58,000 infected, 3,000, 5% dead, and other consequences. This is a picture from one of the New York hospitals. This is the other consequence, the iron lung. I remember when I grew up, that was quite common when I was a kid. Iron lungs, you knew people, or you knew of people that were there. There were no vaccines in 1952. All right, the other thing was hysteria. This is a town outside of New York City. People were putting up signs preventing free movement. Children under 16 not allowed to enter this town, and there are police enforcing that. Now, polio wasn't just a disease of children. It affected adults too. Most famous was Franklin Roosevelt. He contracted polio in 1921 at the age of 39. So it wasn't just a children's disease. It affected everybody, mostly children, but everybody was at real risk. Now, there may be, I'm not going to show a show of hands, but there may be some survivors in this room, I don't know, who are post-polio syndrome victims. So Post-polio syndrome is a condition that affects people who had polio, recovered. It causes muscle weakness, fatigue, and it worsens over time. It's not contagious, but it usually develops 15 to 40 years after the initial infection. So there are many people in this country our age who were children before the vaccine, and they're still suffering the consequences. Measles, complications, pneumonia, seizures, encephalitis, blindness. 1980 worldwide, 2.6 million people died of measles. That's not very long ago. And as Rick was saying, there's an outbreak in West Texas now. Last time I saw 58 people have been, I don't know if they've been hospitalized, but certain 58 people have cases. That's likely to explode. To be 58, I think it's gonna, by next month, you're gonna have a lot more than 58. It's where it's spread to New Mexico now, and that has the potential to be a real problem. So I mentioned that. Let's see. Diphtheria. Diphtheria is a serious bacterial infection, affects mucous membranes, causes breathing and swallowing problems, and it can be deadly. 1921, that was before the vaccine, 206,000 cases in the United States, 7.5% fatal. So it's not something to laugh at. Diphtheria is a serious disease. Fact of life. Before vaccines, ubiquitous, legally enforced quarantines. So if you had a case of diphtheria in your house, somebody from the health department would come, staple that sign to your door, or if you had polio, somebody in the house, people in the house were forbidden to leave the house, people were forbidden to enter the house. This was common. Talk about loss of freedom of movement. That's legally enforced quarantines. That was a fact of life. Now, COVID-19. Somehow there seems to be amnesia in this country, in my opinion here. This is a makeshift morgue, Belleville Hospital. So many people were dying, there was not room in the morgue. These are refrigerator trucks lined up in the parking lot to store the corpses. I don't understand how people sort of forgot what was going on. It wasn't very long ago. It wasn't just New York. Went coast to coast. This is makeshift morgue in El Paso, Texas. Same thing in Los Angeles, San Francisco. Every major city ran out of morgue space. Had to take refrigerated trailer trucks to store the corpses until they could be buried. Okay, that's enough of that. That's the ugly. The good. What the world looks like in the presence of vaccines. All right, 1947 smallpox scare in New York City. Now, that was before I was born, but I remember I was from New York, and I remember my mother telling me about this. What happened was somebody came from Mexico City to New York on a plane, and he had active smallpox. So there was a smallpox scare. 6.3 million people were vaccinated over generally about a month. This was a picture you can see of individuals lined up to get vaccinations. That's how people in 1947 viewed vaccinations, far different from today. But again, smallpox was smallpox. Now, the World Health Organization launched a plan to eradicate smallpox in 1967. They did that by widespread immunization and surveillance around the world for several years. The last known natural case was in Somalia in 1977. World Health Organization declared smallpox eradicated in 1980. Now, smallpox is the only virus that has ever been eradicated over human history. No other virus. Every other virus, they're still circulating in the environment. The only samples, presumably of smallpox, are samples that the United States have, I believe, in Bethesda, Maryland, and Russia has probably in St. Petersburg or Moscow. They're supposed to be the only samples. Other than that, smallpox has disappeared from the planet. And they did it by vaccinations and the World Health Organization. I should mention the United States is withdrawing from the World Health Organization. Okay, now, Salk vaccine, 1955. That's kids being immunized in the school gymnasium. I remember being immunized in my school gymnasium, and I suspect most people here remember that as well. But that's how my first remembrance of vaccines was the 1955 lining up in my elementary school gymnasium to get vaccinated. This is the impact from the CD, I didn't think it was CDC, of vaccination on paralytic polio cases and deaths in the United States. Of course, this was the peak of the 1952 epidemic. This was where the mass vaccinations in the United States started. That's what vaccines did for polio. Now, I should also mention at this point that there were actually 3 strains of polio. 2 strains have been eliminated from the planet, like smallpox. 1 strain is still endemic. Mostly that's centered on Afghanistan and Pakistan, that region where they share a border. But that's where polio, it's in other places, but that's the primary place. That's still there, and it could leave. So polio is still a problem. This just shows the impact of vaccination. This was from the UK, but the results were similar in the United States. So if you look at all these diseases, diphtheria, can't see here, measles,
influenza,
whatever. Well, I think polio's here too. That one here. I can't read which one, but you can see the point is where the arrows are is where the immunizations start, and you get the same result. Before the immunizations, prevalent disease. After the immunization, essentially 0 for all intents and purposes. What's particularly relevant these days is measles, I guess. Kids aren't. This is where we were, where we have been, and we're going backwards now. And that's because people aren't being vaccinated. Now, this is the impact of COVID-19 vaccination on U.S. death rates. This was beginning in 2021. These are unvaccinated individuals. Blue line is vaccinated individuals. And just for a point of reference, is the number of people that were killed in the World Trade Center was about 2,800 or something like that. We were losing over 8,000 people a week with COVID. And this is what the difference with vaccines. So this is just a fact. Okay, now the bad. No pharmaceutical, including vaccines, can be absolutely 100% safe for all individuals. There can be serious side effects from vaccinations. And the rest of this talk is a very brief introduction to the immune system, because in order to understand why there are side effects and put that into perspective, one needs an understanding how the immune system works and how vaccines work or they don't work. So unfortunately, I can't give you a immunology talk that would explain that all in 1, 2, or even 3, 4, 5 hours. I have to spend the whole semester talking to you about this. So what I'm going to try and do, well, the idea is I'm trying to give you the view from 30,000 feet. Okay, so you can imagine taking off from Los Angeles going to New York. So the first thing you're going to see is maybe the high Sierras. Well, we'll try talking about that. Then maybe you'll see Salt Lake, and then the Rockies, and you go over most of the rest of the country. You don't see very much. If you went down, there's lots going on, but you don't see it. You get to New York, you see the buildings. So what I'm going to try and do is give you that view from 30,000 feet. And it's going to be incomplete, and I'm sure there are going to be questions, but that's unavoidable. Even so, I rather suspect that a lot of people are going to feel lost. Don't worry about it. I'd be surprised if I hope that you get it, but I really don't know. My experience is teaching physicians and medical students, and this is, to be honest, my first attempt to talk to the general public. So what I'm trying to do, is make this at the level of a scientific American. And if I succeed, we'll see. But that's the idea. So if you start seeing science and your head starts spinning, don't worry about it. It's not you. I didn't do as good a job as I wanted to do. All right. First thing is, what's the immune system? It actually turns out that there are two kinds of immune system. There's what's called innate immunity and adaptive immunity. Think about the innate response. That's a rapid response. It takes place in minutes. That's not what vaccines are really about. And there are certain cells, and the response isn't well targeted. And there's a lot of collateral damage. I'll talk to that in about 1 second. The adaptive immunity is really what we think about, most people think about when they think of immunology. That's a slow response. You think if you get a cold or infection, it takes a couple of days to start responding. That's your adaptive immune response starting to kick in. The cells are, we'll talk about the cells that are involved. It's highly specific. So if you have a sore throat or something, the immune system's going to target the sore throat, but it's not going to be doing anything to your big toe. So it's a targeted kind of response. And the other part is that the immune system, the whole point is it has memory. That's what vaccines are all about. So if you're immunized or you're exposed to some pathogen, the second time you're exposed, you do better because the immune system, hey, I know this guy, I've got to get moving quickly. That's what the adaptive, that's why it's adaptive. So it learns from past experience, learns history. Now, actually they're kind of connected, there's some interconnections, but we don't have time to talk about that. Innate immunity developed very early in evolution, so this is a sea urchin, so it has an innate immune response. If you think about innate immune responses, when you get a splinter, it gets red, it hurts, that's your innate immune system kicking in. So your blood vessels swell, you get cells called neutrophils that are going in there, it hurts for a while, but then it clears up. That's the innate immune system. Don't have time to get into that. So that's all I'm going to say really about the innate immune system. Adaptive immune system, on the other hand, protects against viruses, parasites, bacteria, and cancer. That actually was controversial for a long time, through the 1950s, 70s. That's come to fruition. So vaccines and immunotherapy are now actually the big advances over the last, I'd say, 10 years or so, have been the targeting of the immune system for cancer. And all these ads you see about all these drugs for cancer, they are all vaccines. They're not vaccines, but they're agents for immunotherapy that enable the immune system to cure cancer. I'm not going to talk about that. There are research projects that are in fact aimed at developing vaccines to prevent cancer. Again, that's not what we're talking about today. What I'm talking about today is vaccines for viruses and bacteria. In both cases, what the vaccines do, the vaccines don't work independently. The vaccines are agents that enhance your immune system to take care or to combat the potential pathogen. Now, the immune system is actually, it's not like the heart, it's distributed all through your body. There are various places where this happens. So there are tonsils, there are in the kidney, the appendix, there's two special ones. There's the bone marrow and the thymus. The bone marrow and thymus are very different from the other organs like lymph nodes. The bone marrow and thymus are where immature lymphocytes are, and the cells then migrate out. I'll talk about that later, and to become mature and actually function. But the point I'm trying to make here is the immune system is distributed out throughout your entire body. It's not limited to any one spot. All right, so progress in understanding the science behind adaptive immunity has actually historically been connected to immunizations and vaccines. So the vaccines and the study of immunology have actually developed hand in hand. Now, this is interesting what I found. The first mass immunization in the United States goes back to the founding of the Republic. George Washington ordered the mass immunization, at this point it was called variolation, to protect against smallpox and the British Army for all troops in 1777. Now, he did this, actually, the Continental Congress told him not to do it. He ignored them and did it anyway. Now, I say the British Army because the British Army were in fact employing biological warfare at the time. They were taking infected, smallpox infected individuals, crossing the American lines and infecting American troops at the time. They were able to do that because the strain of smallpox that was prevalent, the British troops were more or less immune to it, but not the American troops. At the time that George Washington ordered this, I think the statistics were something like 40% of his troops at the battalion level were out of commission. Had that continued, the Revolutionary War would have been over in 1777. After the vaccination, at the battalion level, 100% of the troops were ready for combat. That was the difference. And so, by bias, you have all this talk about military enforced vaccinations. If it wasn't for vaccinations, there wouldn't be a United States Army. All right, so the science of immunology actually started with Edward Jenner in 1796. He was a British physician, and he showed that if he could vaccinate children or individuals with cowpox. Cowpox was a disease that's similar to smallpox, but it wasn't particularly fatal. Milkmaids, women who were in charge or were employed with milking cows, they did all that by hand, tended to get infected from the cowpox. He took the pus from some of the skin eruptions from these women, and he inoculated the first child in 1796. Now, these days you wouldn't get away with doing that kind of experiment. But back then, okay, but it worked. Now, I found this, anti-vaccine conspiracy theories are nothing new. This was a publication right about 1800, and it was from the Anti-Vaccine Society, and the title was The Wonderful Effects of the New Inoculation. And if you look closely, you see it's telling you that people are going to turn into cows. So you have this cow face, and then if you look at some of the arms, there are udders that are popping out of people's arms. So this was the stuff that was, so again, you know, the anti-conspiracy series didn't just start with COVID-19. All right. Now, we're going to start getting into the science. So I hope that if you don't understand something, we'll see, you can raise your hand, maybe I'll stop and slow down. But the first part of this, vaccines antibodies were discovered in 1890. And these two Europeans, Burley and a Japanese, discovered, they were working with diphtheria, and they discovered that that the vaccine worked by producing something in the blood that they termed antibodies. Now, they didn't know what antibodies were. They couldn't see it. They didn't chemically. They didn't know what it was, but they knew that something was there that that was produced when they inoculated. They were using guinea pigs at the time, so I'm not going to go through if I was talking to medical students, I go through the experiment. But the point was that if they took cholera toxin from an infected animal, inoculated the blood from the infected animal to another animal, that animal was then protected from the cholera toxin. If they had not, and it was strain specific, so it had to be the right strain. So if you immunized with strain A and infected with strain B, you'd still get it. So you had to be immunized with the right strain. And so you were able to teach the inoculated animal what the toxin, what was there, and protected him. And again, they said, well, this is because something was produced in the body, in the blood of the inoculated animal, and they called it antibodies. It was anti-toxin. The reason that diphtheria is a problem is the bacteria produce a toxin that kills cells. But they could produce something in the blood that neutralized the toxin, so it was an antitoxin or antibody. So this was the first demonstration that such things as antibodies were there. That was 30 years later, in 1921, it was shown that repeat exposure to toxin actually gives a stronger secondary response. So this was the first response, this was the secondary response. So this showed that immunization, again, this was science and the study of immunizations are going hand in hand, and it showed that after the second immunization, you got a much bigger response. That's a characteristic of immunizations. But again, they didn't know what antibodies were. It was just something in the blood called antibodies. Subsequent response showed that the stronger antibody response actually, that the lag phase, there was a lag phase, and so the first time it took, I don't know, say 14 days, 2 weeks to get a maximum response. But the second time, the response was much greater, and the response was much quicker also. So, two things. The system, remember, this response is stronger, and it happens faster. Now, if you've got a bacteria that's growing, you want to start getting rid of that as soon as possible. And that's what the vaccines was doing. So it was a strong response, and it was quicker. That's characteristics of the adaptive immune response. All right. So this was the 1930s. This was how immunizations were understood. You had the capacity to recognize and respond, produce antibodies to almost an unlimited amount of antigens. I didn't go into that. But the point was that the immune system can recognize all sorts of things. Okay, say infinite, but certainly a large number. 2, you had the capacity to distinguish foreign from self. So while the immune system could recognize foreign cells, like bacteria, it was thought that it didn't recognize self-cell. So it had this self-non-self ability to recognize. And 3rd, which is most important, it had the capacity to learn from and remember prior exposures to antigen. That's the basis of immunizations, the ability to learn and remember. So if you don't take anything from this lecture, that's what immunizations are about. It learns and it remembers. So the first time you may get, and this happens to the immune system, so the first time you may get sick, but the second time you're not going to respond as, you'll respond and you won't get as sick. The vaccines are actually designed to make it so you don't get sick the first time you're exposed, but we'll get into that. All right. Now, it's only 1939 that Celius and Cabot was an American, recognized what these antibodies were. Turns out they said antibodies are proteins. And they knew that these proteins, there were a bunch of proteins in the blood, and it would the antibodies belong to what's called the gamma globulin fraction of proteins. So people have known about these proteins in the blood, but they had no idea what they were doing. These guys said, okay, these proteins are what the antibodies are. So in 1939, people understood, up until 1939, antibodies were just these nebulous things. 1939, not nebulous, they're these proteins. All right, so where do these antibodies come from? Well, it was 4 years later, 1943, that they said, well, these antibodies, these are the antibodies. These green things and the red things are what I'm speaking is antigens. These came from certain cells called plasma cells. So they didn't magically appear, they came from certain cells that produced them. So this is a big advance in 1943. This is where antibodies come from, these cells. Now, these cells were first described in 1875. So this is a stained cell, and these are red blood cells to give you an idea of what the size is. Well, these cells, again, these cells have been around, people knew about them, but they didn't know what they were doing. 1943, this is what they're doing. They're producing these antibodies. They're part of the immune system. That was 43. 44, independently, 3 immunologists discovered that, well, there's not just one kind of immunoglobulin antibody, there are actually 2 kinds. There's what we call IgG, and they discovered IgM. I won't get into what the differences are or how they did this, but they said, well, there are 2 kinds of proteins, and they're both antibodies. They both come from these, the plasma cells that we think, but they didn't know. Again, so the point is that they're proteins, but they're not all identical. There are differences. Okay, now after the discovery of IgM, it turned out that, remember I told you there was a secondary response that was different from the primary response. Turned out the secondary response was driven by this second class of antibodies, IgG, whereas IgM was involved in the primary response. So the major protection actually was not so much from the IgM, but rather from the IgG. Okay, now we talk with Jonas Salk. Jonas Salk got the polio vaccine in 1955. The reason that I put this in here is what I've told you about immunology is what Jonas Salk knew about immunology in 1955. So all of what I've introduced to you is as much as Jonas Salk knew at the time that he produced this polio vaccine. Everything that I'm going to tell you from here on in was unknown to Jonas Salk in 1955. And I found that, kind of astounding, because it showed you how important the polio vaccine is, and they were able to do this with this limited knowledge. The science of immunology essentially exploded from 1955 up until now. And I'm going to sort of go really quick, and you'll see what I mean. But what you know now is what Jonas Salk knew when he was working on the polio vaccine. So if you could go back in time. Okay, now, the big advance for how the immune system works was what was called the clonal selection theory, and that was formulated by a Australian, Frank Burnett, 1957. Now, I circled this slide in red. The reason I've done that is when you leave here, if you don't remember anything that I say about the science of immunology, remember this slide. Because this slide essentially tells you how the immune system works. Now, when he did this was theoretical. The proof wasn't until a few years later. At any rate, the way that the immune system works is that he envisioned that there were a bunch of cells, and these cells had on their surface antibodies. So he knew about antibodies, he knew there were proteins at this point. And he said, well, there are antibodies, but each cell, the antibody, has a different shape. So different cells make different shaped antibodies. And what we're talking about, the shape is really important because the shape is complementary to the antigen. So it's like a lock and key. So this is representative of one shape and one antibody. So this antibody recognizes this shape. This antibody does not recognize that shape. And the idea was that these antibodies appear at random on the surface of all of these cells. All of these cells have different shapes, so they can recognize different antigens. Now, if the shape is just right, the antigen comes in and then selects the cell. So this antigen selects this cell. The implication of that is that information, this binding information, is transmitted into the cell, and this cell begins to divide and expand clonally. This cell doesn't divide because there's no antigen binding on the surface here. That's regular. It's all about shapes. So this shape is incompatible with the antigen. So you can imagine this is a antigen from a bacteria or a vaccine. These cells are going to expand, and that's what's called clonal expansion. Then these cells differentiate, and at that point he knew that these were plasma cells, and they begin to secrete antibodies. So it's no longer antibodies that's bound to the surface, but these guys produce the antibodies that are secreted. And the point is, these antibodies have the same shape as the ones on the surface, so they essentially eat up all of the, or bind to all of the antigen. You can think of the antigen as bacteria. All right, so this is the clonal selection theory. That is the fundamental understanding of immunology. That's what immunology is all about, in essence. And so lots of questions. Well, what's this? How's this information get transmitted? What are these cells? That's all icing on the cake. And that's the area where I was working in, but we won't get into that. At any rate, a corollary of the original clonal selection theory was that there must be non-self recognition and tolerance to self-antigens. So in other words, suppose these are two antigens. Well, this cell here has, and suppose this is a foreign antigen, well that goes as we said, but there must be some mechanism, because if these shapes are random, well, you're going to end up with antibodies that can, on the surface, they can recognize self-antigens. So the original clonal selection theory was that there was some mechanism here that prevented this cell from expanding. And that, I hope I get into that, that's an active area of investigation. How does that work? And that has a lot to do with vaccine side effects. At any rate, the discovery of autoimmunity, and in 1959, two scientists, and they were at the University of Buffalo at the time, Noah Rose and his mentor, Ernst Witebsky, actually showed that, well, that's not quite right, that the immune system actually does make antibodies to self. They did that in, rabbits, I believe, rabbits and thymus. And they could show that they could produce antibodies in rabbits to their own thymus tissue. That's called autoimmunity. And that was a big deal at the time, and nobody believed it. It was hard to get that, it took a while for that to sink in. But the fact is, that's real. So in fact, you do make antibodies to the self. Now, that's the basis of autoimmune disease. And that's when the controls are out of whack, and you begin to make too many self-antibodies. And so there are now something like 80 to 150 specifically designated autoimmune diseases, and some of them are very specific, like Hashimoto's thyroiditis, that just affects the thyroid gland, and all the way down to SLE, systemic lupus. And lupus can affect any organ. Somewhere in here is arthritis, and so forth. So, this is a fact in life, is autoimmune disease. And that's because the immune system begins to attack self-tissues. Now, next, going historically, people knew that the antibodies were immunoglobulins, and they knew that they were proteins, but they didn't know what the shape of these proteins are. I mean, these are molecules, you just couldn't take a microscope at. And I told you the shape is very important. So, in 1960, they won the Nobel Prize for this. I should also say that there was a Nobel Prize awarded for the clonal selection theory. And so a lot of the things that I'm talking about here are highlights of people who won Nobel Prizes for this work. Edelman and Porter won the Nobel Prize, I think it was 1980 or something, when they determined the structure, and by structure, I mean shape, of what the immunoglobulin molecule looks like. So this is what the structure looks like, or the shape, and turns out that it's like a Y-type kind of shape. So actually, you see a lot of publications, antibodies have this Y shape, and this is from this work in 1960. Now you have to understand that they couldn't see any of this. This was all done by biochemistry, and they figured out that in order to explain their biochemical results, this had to be the shape. I won't get into the kind of experiments. And anyway, it turns out that there were the immunoglobulin molecule had these three regions. These two regions were called FAB, and this was called FC. And the reason they called this FC because they had trouble isolating this because it crystallized all the time. So that's why they called it FC, F for crystallization. At any rate, they turned out this Y shape, and they were binding antigen at the end of these arms, and then these things had a little, they called a hinge region, and they were flexible. And again, they determined that all this was just biochemistry experiments. Now, let me go back a second, and I've talked about proteins, So what are proteins? Well, proteins are strings of amino acids that fold up to give a unique structure or shape that's determined by the primary acid sequence, amino acids. So these are amino acids. There are 20 different possible amino acids. And a protein is essentially a linear strand of amino acids. Each amino acid has a different composition that's determined by the gene. So each gene codes for a strand of amino acids. I'm not losing anybody here, am I? Am I?
Nope, go for it.
All right, so you think of it like beads. So that's what protein is. It's a linear string of beads. And each bead is what's called an amino acid. So you don't have to know what amino acids are, but just think of them as strings on a bead. That's what proteins are. Now, the point with proteins is that they don't remain as strings, they fold up. That's what this is. And so this is the string of beads, and these beads fold up. This bead is what gives this, the way that this folds is what gives the protein its shape. Each protein folds differently. Now, turns out that proteins may be composed of one or more of these chains. So this is one sort of string, is red, this dark green is another string, and this third light green is a third string. So these strings all fold up, and it's not just an incoherent knot, they actually have real structures. And this is how you would visualize the way these strings fold, so this is a space fillet. So this is the red string, it has this shape in 3 dimensions. This is the dark green, and this is the light green. So these fit together just right. And that's why, so these individual strings are called subunits. So they're part of a protein, but most proteins are a combination of these strings. And again, it looks like this, when you look at it like this, it looks like a knotted mess, but it's not. They have very specific structures, and these structures give very specific shapes. Okay? So you end up, you start with strings, they fold up, but they don't fold up at random. There are real shapes to them. That's what proteins are. Okay, so this is what the antibody looks like. The immunoglobulins, they actually have 4 strings. What they call a light chain. Now these are identical, so there are 2 light chains and 2 heavy chains. So the light chain folds up like that on that arm. The light chain folds up like, heavy chain folds up like this, and they intertwine and they make a shape. And this is the shape with the 2 light chains intertwined together. Well, this looks very much like the picture I showed you. And this is what so this is more like what a real immunoglobulin molecule looks like. But this came several years afterwards, this kind of picture. But nevertheless, that picture was right. Over the years, this picture has been upgraded, so we still have the same model, basically from 1960. And so these are the green are the 2 heavy chains. Yellow is the light chains. And it turns out that, where I said that remember I told you 2 kinds of immunoglobulins? Turns out there are actually more than 2, which were discovered by 1970, and there are actually 5 different immunoglobulin chains. So the heavy chain can be one of 5 different genes, and that's how you get IgE, which is important for allergy, IgA, which is what mucosal immunity, IgD, IgG, and IgM, and I won't get, you don't have to remember all of that. But the point is, it's more than just one kind of immunoglobulin. But the important part here is that these regions, both of the heavy chain and the light chain, each what are called, these are regions of the molecule that are variable. This allows the, I'll talk about this in a while, how the immunoglobulins can recognize and bind to different things. So the shape of each of these changes. Now, here is actually a picture. We can now have, like 1960, they couldn't do this. This is a recent picture of an electron microscope looking at immunoglobulin molecules. What does that look like? Looks like that Y. So that's what it really looks like. Now, this is even a more detailed picture, which was reconstructed from what we call X-ray diffraction. Here you can see the individual chains. Remember I told you I had that hinge region? Well, that's a really thin, that's where the beads are actually opened up. You got these beads. So these guys determine the structure, and now, if we had this technology, we wouldn't have to do all the biochemical experiments. We'd go right to this kind of analysis. But these days, you can see this, 1960, Porter and Engelman couldn't see this. But we now have the technology where you can actually see what these molecules look like. Turns out they were right. Okay, next, 64, the question is, we had plasma cells secreting these proteins in the globulin. Where'd they come from? Well, Moore and Schomburg said, well, they actually come from what are called lymphocytes. And lymphocytes bind to have this immunoglobulins on their surface, and they produce the plasma cells. 1964 and 64, people didn't know that. So lymphocytes were actually first described in 1890. This is a lymphocyte, and these are red blood cells. So it's a little larger, has a nucleus, these guys don't. But this is what a lymphocyte looks like. And this is the comparison of a lymphocyte to a plasma cell. Plasma cell is slightly larger. And there's a lot more cytoplasm in the plasma cell. This is where actually all the machinery for producing the antibodies are. At any rate, this was a picture that's still pretty much right of how the antibody response takes place in about 1965. And the first part was that you have stem cells, and then these differentiate into this uncommitted kind of cell, and then these form immature lymphocytes. And the point here is these lymphocytes all have immunoglobulin on their surface, and all of them have different shapes so they can bind different antigens. This is where the different shape part comes from. Now, the antibodies, some of these shapes correspond to self. So these shapes were thought not to respond, and the shapes that were foreign were allowed to mature, and then if they had the right antibodies, so this cell has a shape corresponding to antigen 3. This cell has a shape that corresponds to antigen 4. This has a shape that corresponds to antigen 1, so when they bind, they produce antibodies. The point is, there's no antigen 1 here, so this cell doesn't do anything. Only the shapes that actually bind something are allowed to proliferate. So that's nothing more than I said before. But the point is that now we understand that there actually are a pool of these cells that are produced, that respond to cells. They don't disappear. And they undergo a process called tolerance. And for the most part, they don't produce plasma cells that then give us a problem. But this process can be interfered with by genetic abnormalities, environmental agents, including bacteria, viruses, and vaccines. That's the origin of autoimmune disease. So the origin of autoimmune disease is when these mechanisms that prevent cells that have the ability to recognize self are allowed to progress. Originally, they're supposed to be held in check, but sometimes those mechanisms fail. It's not clear, it's an active investigation, I'll get into that later. Active investigations, sometimes they fail. Again, the agents can be environmental agents, like pesticides or heavy metals, things like that, or a bacteria or virus infection, and vaccines. So when things go south with vaccines, very often the problem is that these tolerance mechanisms have been disrupted. Okay, so vaccines and all, but the point is that vaccines don't do things differently really than the viruses or bacteria that they're designed against. Okay, all right, the next highlight in immunology is the discovery of T and B cells and their development and origin. This was Good and Cooper. They were at the University of Minnesota at the time, 1965. Cooper's still alive. Robert Good passed away in 2003. The point here is two things. One, as I talked about lymphocytes. Turns out that there are two basic kinds of lymphocytes. There are what we call B cells. That's what I've been essentially talking about. B cells produce antibodies that are involved in that. But then there are T cells. Turns out that the guys doing cancer vaccines are all interested in T cells. So some of you may have heard about T cells and B cells, but this was 1965, was the developmental origin. So I've got this one in red as well, if you can notice it. And so this is the second thing that you should remember. Aside from clonal selection theory, think that there are T cells and B cells, two basic kinds of lymphocytes. All right, I'm going to finish this in a second. All right, all right, so the point of this is that there are two kinds of cells, T cells and B cells, and the T cells develop in an organ that is called the thymus. That's an organ right over here. And the B cells for humans actually develop in the bone marrow, the immature cells. This is where they come from. Now, I remember when I was a kid that people used to be irradiated for what the physicians called thymuses that were too large. Turns out that was a mistake. Thymuses are there for a purpose. They're there to produce T cells. As you age, the thymus decreases in size, it atrophies. So the thymus, so most people here, the thymus is very small. At puberty is where it's at maximum. One of the theories as to why the immune system gets weaker over age is because the thymus decreases and you have less ability to produce these cells. Again, I'm not sure they agree completely with that, but that's a prevalent notion. Okay, I'm going to go for just about 2 minutes and then we'll take a break. I'm used to lecturing for 3 hours at a time, so. All right, so I want to look at a closer look at B cell activation and differentiation. So we start with the virus. The virus produces antigen. The B cell then recognizes that antigen. And this is the point here. When the B cell recognizes, so the antigen on the receptor, when the immunoglobulin is on the surface of the B cell, it's called the B cell receptor. And its function is to recognize antigen and signal the B cell, hey, something's out here. B cell then begins to proliferate. And the point is that it doesn't just produce plasma cells, it also produces what are called memory B cells. This is the fundamental basis of the ability of the immune system to learn. That's why the secondary response is bigger and faster. So The first time you've seen an antigen, you have to go through this process. Second time it's around, you have a number of these guys that are already there. It's like when you're infected with a pathogen, it's a race. If you're infected with bacteria, those guys are doubling every 20 minutes to an hour. Your cells take maybe 18 hours to 24 hours to double. So they're running a lot faster than you are. And your immune system has to catch up. And that's why you can get really sick before you produce enough cells to fight an infection off. By having these memory cells, it's like you're starting, if you're going across a football field, you're starting at the 50-yard line already, where they're starting at the opposite goal line. So you've got a head start. That's the point of having immunizations, is to produce these memory cells. Okay? And then the memory cells can then immediately start producing plasma cells that produce antibodies. All right, quickly, we talked about the antibodies can be either soluble, and that's because there's a little difference in the terminal over here. All right, I'll stop over here. All right, so we can take a break, and we'll pick it up in a little while. Now, is anybody completely lost? Well, like I said, it's okay, and I'm here for questions. But again, I'm just trying to, like I said, this is really a 30,000, for each slide I could spend an hour talking about that. And that's obviously not right. But there are some things that I want to at least get through so you can at least understand how the immune system is supposed to work, and then we'll talk how vaccines interact with that. Okay? Okay, I guess everybody is ready for some more?
Yeah.
Okay. All right, I just had a woman come up and she said, when are you going to talk about natural immunity and, as opposed from vaccines? Well, the answer is essentially all I've been talking about so far is really natural immunity, is how the immune system, I haven't really talked about how vaccines come into the picture. I've talked about pathogens, bacteria, and so forth. So essentially what I'm doing now is telling you what natural immunity is and how the immune system works naturally. And I will then, in a little while, talk about vaccines, how vaccines enter the picture, and you'll see that they do the same thing as in natural immunity. So that was a good question. Okay. Now, this is very quick, and so for the next 3 or 4 slides, if things start spinning your head, forget about it, and we'll come back to more, we'll get grounded more quickly. But I'm going to start this slide here with what's called the central dogma of molecular biology. And if you go to take a high school biology class these days, they'll probably talk about this. So this really started with the discovery of Watson and Crick in 1954, when they discovered the structure of DNA and that DNA are genes. And so the central dogma of biology says you start with DNA, which is genes, and then this is a sequence of letters, which are what they call nucleotide bases. So forget about what they are, just think of an ATC, linear sequence. This gets transcribed into what's called RNA. I'm sure everybody's heard of DNA and RNA, particularly the RNA vaccine these days. And so you get RNA, and then the RNA gets translated into the protein, to the peptide, which is what I was talking about before. So you go from the gene to this linear sequence of amino acids, which is a protein. This is the central dogma, what's called the central dogma of biology. So the central dogma of biology led to what this question called the generation of diversity, or immunologists used to say the God problem. And the question there is, how is it that lymphocytes can encode enough information, have enough genes, so as to produce the number of diverse immunoglobulin protein that as a group will be able to recognize and bind to virtually anything in nature. So I just told you that these immunoglobulins have these 5 shapes that are determined by the sequence, but they, thousands of sequences, we can respond to. How does that happen? How does it fit into the gene? How can you have enough genes to do that? And the answer is partly the discovery of what was called VDJ recombination. This is 1976, right? So we started, so the structure of immunoglobulin was 60, this is a little later. Tanagawa, and again, this was another Nobel Prize. And turns out that these polypeptides or proteins are actually in the DNA. There's not one sequence, but there are a group of sequences. So they're split up. So there are a number of V sequences, a number of J sequences, and you pick one at random to join another one at random. And same thing for the heavy chain, except now you've got 3 kinds of sequences, so that you have a whole lot of them. So instead of, so one person can have one, another person can pick another, and essentially it's like a slot machine. So this is the VDJ, you pick a V, you pick a D, and you pick a J. And so one piece of DNA can code for literally, I think the number is like 10,000 different possible combinations. So the point is that even if you have a genetic twin, his immune system is going to be different from yours because of this randomness to it. And that's the way that we can, one of the ways that we can, there's even more randomness, but I'm not going to get into that. Turns out that this can lead to something like 16,000 different combinations with the number of genes that we have. But then there's also random mutations in there as well. I did the calculation, it turns out you can get 10 to the 160th possible combinations, which is more than the number of atoms in the universe. So that's how we deal with how the immune system deals with it. This is very important. All right, so turns out that we said that there were T cells and B cells. We said that when the globulin was on the surface of the cell, it was a receptor, and that's different from the secreted variety. Turns out that the T cell also has something. The genes are different, but structurally it looks pretty much like the immunoglobulin proteins. And they use the same kind of mechanism to get this diversity. All right, have I lost anybody yet?
A little bit.
Okay. Okay, now, we'll move on. Turns out that things get even more complicated. They're not just T cells and B cells. Turns out there's a different kind of T cells. And turns out that there are what are called cytotoxic T cells, which are, call them CD8 because that's a molecule that's on the surface. And there's another kind of T cell called CD4. CD8 T cells are kind of T cells that are actually designed to destroy cells. So they're killer cells. The CD4 cells are what are called helper cells. So when a B cell responds to an antigen, most of the time it requires, turns out that it requires an interaction with a helper T cell. So this interaction with antigen most of the time is not enough. It needs the second help, and then you can get the plasma cells formed. The CD8, this is a what's responsible for what's called cellular immunity. So the idea is what these cells do, if you have a cell that's already infected with a virus, the immune system is designed to get rid of that virus by killing the infected cells. Get the picture? Mm-hmm. So that's the way the immune system really works. The idea is once you're infected, to get rid of that infection, you can't wait till the pathogen or the virus or the bacteria is in the system, you go right to the source, and that means you kill the infected cell. That's a really important concept. And that's why when you get sick, most of the time, or a lot of the time, you're feeling sick not because the pathogen is killing your cells, it's the immune system that's killing your cells. Okay? Sort of like what happened in Vietnam. You had a village that they were infested with the Viet Cong. Well, we're going to wipe out the whole village. That's how the immune system works. If you have an infected cell, the immune system is designed to destroy that cell. And that's why you have side effects and why you feel particularly sick. So most people think that the immune system is designed to prevent you from getting sick. Turns out not so much. It's designed, once you're infected, to get rid of the pathogen. And that's the function of these CD8 cells. And these CD8 cells, what I talked about a little earlier with the guys looking at anti-cancer, these are the cells that they're trying to stimulate to get rid of cancer cells. So cancer cells are cells, but these you want to so these cells will recognize and destroy cancer cells, but sometimes you need to give them a little help. That's what all this immunotherapy is all about, is helping these CD8 cells recognize a cancer cell as something that has to be destroyed. But they're again designed to destroy infected cells. Okay, so this is a simplified overview, and I say simplified. So each of these actually, I can expand this by 20 times now. And so this really is a simplified view. So you have a common, the immunoglobulin, positive cells, B cells, can form plasma cells or memory cells. Then you have T cells. Well, the T cells can form this arm, the CD8 cells, or it can form CD4 cells. Again, these are the helper cells. These are the cytotoxic cells. We have memory cells in both arms. So the cytotoxic T cells and the memory cells also have this capacity to memorize, to have cells. Remember, the reason for that is if you get infected, you're starting at the 50-yard line. You're not going to go through all this stuff to get to where you need to be. So this is built into the whole ethos of the immune system. And again, so it turns out if I were to give you a picture, this would be 10 times the size now, because there are actually many more kinds of T cells. Even B cells. Turns out that there's not just one kind of B cell, but there are 4, 5, or 6 different kinds of B cells. And I'm going to get into that now. Again, I could spend the whole lecture talking about B. Yeah.
So under a microscope, can you actually see and differentiate from one type of cell to another?
The answer is basically no. So if you take a regular white microscope, you can't. However, the big advance was the invention of what are called monoclonal antibodies. So these cells have different molecules on their surface. So you can't look at a light microscope and say, hey, this is a T cell and this is a B cell. However, you can have an antibody that recognizes a protein that's only on B cells or a protein that's only on T cells, and then you can put a fluorescent tag on that microscope, and then only the B cells light up, or only the T cells light up. Monoclonal antibodies were developed in 1979, 1980, so that's what made all of this possible, is what I'm showing you here, all of this stuff became possible with the technology of monoclonal antibodies. I'm not going to talk, I can talk about monoclonal antibodies, but monoclonal antibodies were developed. There are actually all these drugs now of monoclonal antibodies that you see advertised on TV. This is MAB, monoclonal antibody, at the end of the drug. At any rate, so the answer is yes, but we can get around that. Okay, now I want to talk about binding. So antibodies can recognize congruent shapes. So this is a cartoon. So this kind of antibody might have essentially a hole to recognize something that's spherical. This could be cylindrical. This is this kind of shape, and sometimes it's a protruded shape. So this is how antibodies work. They recognize in 3 dimensions different shapes. And they can do that because of their shape. And this interaction takes place at the arm. So if this is an antigen, this is the antigen. They call this, you don't need to remember this, it's called a paratope. But the point is, this shape is complementary to that shape, and that's how antibodies can bind to and recognize antigens, this shape. Soluble antibodies can bind to soluble antigens. So if this were a toxin, and this is a host cell, the host cell may have toxins on the surface, which is going to kill the cell, but antibodies can recognize the toxin on the surface, and it can also recognize antibodies, toxins that are in solution. So it doesn't matter whether they're bound to a surface or they're in solution. It's just that shape. And so his antibody is recognizing shapes that are bound to the surface of the cell. So this is pretty straightforward. So an antigen on a surface can be considered a mosaic of different little shapes, you know. And so this B cell has a shape that recognizes antigen 7. And there are other antigens, but it's only antigen 7 that it recognizes. Now, some of these shapes are recognized by, some of these antigens are recognized by T cells.
Question?
Yeah.
What is the recognition process?
Recognition of what?
Is it random?
The recognition process here, again, this was random. So if it happens to have the right shape, it binds.
Just random floating around?
Just random. That's what I was saying. It's, you know, the shape is determined genetically, this random shape. And if it turns out that this shape corresponds to something in the environment, it responds. So this shape picks out the cells that are going to respond to it.
You say it picks out. How does it pick out?
Well, the cells, if you can imagine this as a bacteria site, it's got all sorts of different shapes on its surface. Cells are bouncing around all the time.
It's a random interaction.
Right, so it's a random interaction. Okay? And if the shape is just right, it sticks. Now, I could spend actually an hour talking about that, or more than an hour, but that has a lot to do with what, you know, this issue with masks and all that stuff. That directly affects the question that you're asking. And maybe for another day, we'll talk about that, the biophysics of what's going on here. Answer your question?
Yes, thank you.
Good. Okay, now, T cell receptors. Now, this may have your head spinning because it had immunology head spinning for decades. Well, maybe not decades, but years. T cell receptors don't bind to soluble antigen. They only bind to antigen that's on the surface of cells. So that's a very different kind of mechanism than the B cell receptors. So this is an electron micrograph, a scanning electron micrograph. These are T cells. And this is the surface of a cell that they're binding to. They're binding to antigens on the cell. Some T cells are round, but it turns out that lymphocytes actually are amoeboid, and they don't always have round shapes. Again, that's for another day.
Do they change shapes?
Yes. Actually, when they're moving, they change shapes. Again, that's a lecture for another day. Now, so unlike the B cell receptor, T cells don't bind directly to the antigen. So even though this is a T cell antigen, this won't bind to it when it's, even if it's on the surface. Turns out, I'm going to go through all this, that T cells actually bind to proteins that are unraveled. So remember I told you proteins fold up? What T cells recognize is the unraveled protein. And they actually run pieces of it, which are called peptides. So a protein may be 1,000 amino acids. T cells recognize maybe 10 amino acids. And it's a lineage, so they recognize an unraveled protein. They don't look at the same, the B cells look at the shape when it's folded up. The T cells actually recognize the unraveled protein. In order to get unraveled, the proteins have to be metabolized. So every protein, so if you have a, I'm not going to show this, I'll put it to the side, but if you have every protein in your body gets metabolized and pieces of it get displayed on the surface. So the immune system scans everything that's going on inside your cell by looking at these unraveled proteins. So if you have hemoglobin, for instance, some of that hemoglobin gets metabolized, put into on the surface of the cell in the shape of peptides. That's how cells can recognize cancer, how the immune system recognizes cancer cells. And sometimes there are mutations. The immune system can pick up those mutations by looking inside to it. So what's happening inside the cell comes to the surface. The T cells recognize that. All right, that's really important. That wasn't understood for a long time. Now, not only cell cells proteins, but viral proteins, for instance. Suppose a virus infects the cell. Well, part of that virus gets metabolized, and pieces of those virus get unraveled and displayed on the surface too. So the T cells recognize, hey, this cell is infected with a virus. And then they bring those cytotoxic T cells and they kill the cell. That's how they recognize it. Okay? That's very different from the B cells working. Am I clear? This is actually a pretty important point. So that is why it's so important. The discovery of T cells was so important. Now, the binding of a T cell to an antigen actually is a two-step process. This is even more bizarre. Antibodies just bind to things. Turns out T cells don't just bind to things, and this has consequences with autoimmunity and all this other stuff. So this is, again, really important. Turns out that on the surface of your cells, these antigens that I talked about first have to bind to what are called MHC molecules. MHC stands for major histocompatibility. Physicians recognize these antigens. These are the antigens that they look for when you have an organ transplant. So if you have a kidney transplant, they look for these guys. There they weren't, so everybody, there are a really diverse number in the genome of MHC molecules possible. So there are, 40, 50 different for each, whatever it is, there are thousands of different. That's why it's so difficult for transplantation. But these molecules are not there to make life difficult for a transplantation surgeon. So these were recognized years ago. Only more recently has it been recognized. These are intimate parts of the immune system. They're there to enable T cells to bind antigen. That's what their function is. So the unraveled peptide has a shape to it, and these MHC molecules recognize the shape. Now, step one, the antigen peptide must first bind to an MHC on the cell. The shapes match. So some MHC molecules will match some peptides, but everybody's more or less different. So my MHC molecules may not recognize peptides that yours do. Your peptides may recognize things that I don't. That's one of the reasons why people can be susceptible to some pathogens and other people aren't, because you don't recognize them. And that's genetically encoded. And you'll also think about it, that's why some vaccines work and for some people, but not for other people, because you have different MHC molecules. The first step with this is binding to the MHC molecule. Second step is the T cell receptor. The T cell receptor actually recognizes has a part of it that recognizes the peptide and a part that also recognizes the MHC. So it recognizes the combination. This was a real problem when I started immunology, and it wasn't until mid-1990s that this was worked out. Nobody's won a Nobel Prize for this yet. There are reasons for that, but I won't get into it. But the point is that binding of a TCR antigen is a two-step process. So it's very different from the way immunoglobulins bind things. But it turns out that that has enormous consequences. All right, so this is a picture of again, space filling picture. So this is an MHC molecule, and this is the peptide. So essentially you can look at it, the MHC has a groove, but it just fits certain peptides. Certain peptides fit and certain peptides don't fit. Because the peptides, even though they're not folded up like in a protein, they still have some conformation shape to them. So it can only fit certain shapes. And different MHC molecules fit different shapes. Now, this is actually an original published image, the first published image of a MHC molecule binding a peptide. So this is the MHC empty, and this is the MHC with the peptide that's bound to it. And I remember when I was giving a lecture to immunology students the day before, and I saw this, it changed my whole lecture. You know, just the idea of seeing this, because before this, it was not understood what was going on. It's sort of tragic. The guy that did this, Wiley, committed suicide a few years after he did it. I think he would have won the Nobel Prize had he not. He jumped off at Cincinnati. There's a bridge on I-75 over the Ohio River. He jumped off that bridge. Tragic. At any rate, this picture just shows that there are actually two kinds of I said things just get complicated, and I'm trying to make it as simple as I can. But it turns out that there are actually two kinds of MHC molecules. One, and they call class 1 and class 2. But both of them bind digested peptides. So the class 1, which is what I just showed you, actually binds peptides that have been metabolized by normal cells. So every cell who will metabolize proteins have MHC molecules on the surface. So if you've got an epithelial cell in your throat, for instance, that will express peptides from self, and it will also, if you're infected with a cold or a virus in your throat, it will express that on MHC1. The other ones is MHC2, and that is only special cells that have that. Those are cells that are phagocytes, so those are cells that are designed, part of the immune system that are designed to clear the immune system of pathogens. So some cells, if there are viruses around, or they will digest the viruses and express them on their surface at what are called class 2. But the idea is basically the same. Now, you don't have to look at the details of this, but essentially this is just a overview of the lymphocyte responses, and these are the responses that I was talking about before. This is the B cells producing immunoglobulins, and we call this the humoral immune response. So that's the immune response that we were talking about with antibodies. Well, and all of this is natural immunity. This is how natural immunity works. So this part of the immune system produces antibodies. This part of the immune system produces those cytotoxic T cells. Basically, they work pretty much the same. These depend on MHC class 1, and these both depend on the MHC class 2, but I don't expect you to differentiate. Just remember that the binding is much more complicated than just something smacking onto a surface. And again, this is the way natural immunity works. All right, now, vaccines. Vaccines imitate natural infections. So vaccines work by imitating infection. But they work to engage the body's natural defenses. Vaccines don't work by themselves. Vaccines are just mechanism to enhance your immune system. So if you don't have an immune system that works, all the vaccines in the world aren't going to help you. So vaccines help the body learn to defend from the disease without the danger of a full-blown infection. That's the point of vaccines. So it teaches you about your system, about pathogens, but you don't have to get infected to learn. That's the point. The active ingredient of all vaccines is an antigen, and that's the name for any substance that causes the immune system to produce antibodies. Now, in a vaccine, the antigen can be either a weakened or killed bacteria or viruses. So that's what the polio vaccine was. The polio vaccine that Salk had was a virus that was killed. It was an inactive virus. It was the whole virus that you were inoculated with. Later, the Sabine vaccine was actually a live vaccine, and that was what was called an attenuated vaccine. So that vaccine could actually grow, and if your immune system was not functioning properly, you could become infected with polio from the Sabine vaccine. So in a sense, that was a side effect. Now, it was rare, but in fact, it did happen, and in fact, in some instances, it infected other people. That's why the Sabine vaccine was discontinued. So the Sabine vaccine is not used anywhere for years now. And so the only vaccine for polio is the killed vaccine because there was a danger of the Sabine vaccine. It can also be bits of their exterior surface or genetic material or bacterial toxins treated to make them non-toxic. So the flu vaccine is essentially a bit or a piece of influenza virus that's inoculated into you. The COVID vaccines that came is a little different. They were actually RNA, nucleic acids, and they're injected into muscle, and so you made these peptides. But again, they weren't certainly the whole vaccine, the whole bacteria, but just a piece of the bacteria. So the COVID vaccines could immunize you, and you could have side effects from that, but you didn't get COVID from it. And so I think that's some confusion in the population. So this wasn't, the COVID vaccine is, nobody ever got COVID from the COVID vaccine. That doesn't mean they didn't have other problems, but it wasn't COVID. All right, so this is just a list that I just went through this. The kind of list, these are the currently vaccines that are in use, that can be used. Now, although many different antibodies are produced, the efficacy of the vaccine is usually measured by its ability to stimulate what are known as neutralizing antibodies. So everybody, I'm sure, has heard about that, neutralizing antibodies. Neutralizing antibodies, so if this is a COVID virus, we've all heard about these spike proteins and all that nonsense. I shouldn't say nonsense, but the idea with a COVID virus is that these proteins on the surface of the virus have a right shape so that they fit and they bind what are called ACE2 receptors. These are molecules on the surface of your cells, and the virus gets into the cell by binding to these. Again, it's all about shapes. The right shape fits. So the idea is that antibodies to the vaccine produces antibodies, and some of these antibodies will bind to the spike proteins at just the right point so that they can't, it interferes with it, sterically interferes with it. So this COVID antibody can infect the cell. Now, the vaccine actually produces antibodies that are not neutralizing. So antibodies can produce that bind these spike proteins at places on the spike that doesn't help, that doesn't block. So these antibodies are antibodies against the virus, but they're not what are called neutralizing antibodies. These antibodies are a subset of antibodies that are produced. So when you have a vaccine or a natural infection, you're making antibodies to everything on the surface. Remember? You make a whole slew of antibodies, but only a small minority of them are what are these called neutralizing antibodies. So when the guys that make the vaccines or the physicians that use them determine the efficacy of that vaccine based solely on the ability of that vaccine to produce neutralizing antibodies. Neutralizing antibodies prevent you from getting infected. But the point is that even antibodies that are not neutralizing antibodies protect you. They don't protect you from getting infected, but once you're infected, they protect you. That's one of the reasons why if you've been immunized, those neutralizing antibodies don't stay around very long. So in the case of COVID vaccines, 6 months or something like that. However, other antibodies may stay a little longer, and you've got B cells, memory B cells, and you then produce antibodies that will protect you, not from getting infected, but from getting really sick. And that's why if you get inoculated and you get reinfected, the odds are less that you could end up in a hospital on a ventilator. So, and that's one thing that most people don't, even physicians I think, don't really understand that. That the immune system was really not designed to keep you from getting infected, but rather, once you're infected, to protect you, to destroy the infected cells. So this is a picture of vaccine-induced immunity, and it's exactly the same in the sense of the other picture that I showed you with natural immunity. So this is the virus, this is the vaccine. In this case, it's pieces of the virus. And on this side here, is they're stimulating the CD8 cells, the cell-mediated immunity. Remember I talked about T cells, or these cytotoxic T cells? So this part of the immune response isn't going to stop you from getting infected, but these cells here will find the infected cells and wipe them out. And this is the major cause, major reason why after you've been inoculated and then reinfected, you have less problems than if you weren't inoculated at all. And this prevents you from getting sick. Yeah.
So with COVID, we're getting vaccinated, what, every 6-8 months?
Well, the reason.
What happens, I thought you said these memory cells
Yes.
multiply.
Yes, but the memory cells only come into play once you've been infected. So the reason that you're getting inoculated every 6 months is to keep those neutralizing antibodies around. Okay? So those immunizations that you get will prevent you from getting COVID. However, the immune system, and then so that's sort of just keeps stimulating the immune system to do that. Whereas these memory cells are there to protect you once you've been infected. So they only get activated once you've been infected. So they're not going to stop the infection, but they stop the really bad parts from happening. That make sense?
Sure.
Question, does that mean if at some point you tested positive for COVID-19, you've got the right kind of memory cells?
Yes.
Thank you.
So when people have been tested positive, they're not testing for neutralizers, they're just testing for antibodies. And some of those antibodies may not be neutralizing. So whether you're inoculated with a vaccine or a natural infection, you produce neutralizing antibodies and these other antibodies. So when they test, as you're saying, They just say, hey, you've been exposed. We don't know, it doesn't tell whether you've been exposed by a vaccine or naturally. You just have those antibodies. Now, let me finish with this, because I'm going to come back to that.
Okay.
All right, so this was the humoral. Now, the B cell response, you get the same kind of B cell response as the natural infection, except you don't have to have the disease to do that. And so the point is that what you get is you get the neutralizing antibodies, and the problem with the neutralizing with antibodies are that remember I told you that the antibodies are produced by plasma cells. In most cases, plasma cells have a limited lifetime. That's why your neutralizing antibodies go down fairly quickly. But there's a caveat to that. Turns out that in some cases, these plasma cells migrate to the bone marrow. Those are called long-lived plasma cells. So there are some pathogens that you get immunized, and those plasma cells keep secreting for years. It's not like COVID or the common cold. Sometimes, and I have a question mark there, because nobody knows why that happens. Why some antigens produce plasma cells that end up with long-lived plasma cells, and others don't. COVID is an example of a pathogen that gives rise to plasma cells that are not long-lived. That's why you gotta get damn immunizations every 6 months. This is an area of active investigation now. And so why that happens, I can't tell you, but nobody else will tell you, can either. So this is one of the things that you would want to know with research into infectious diseases. We won't get into the politics of that, but this is one of the things that you want to know. Now, the other thing is that I told you that we had neutralizing antibodies, but it turns out that those other antibodies also protect you. They don't protect you from getting infected, but once you're infected, they do. So, again, we have what are called the other antibodies are able to kill pathogen-infected cells. So they don't prevent the cell from getting infected, but once the cell is infected, they go to work and they kill it, just like the cytotoxic T cells. And they do that by a process called antibody-dependent cytotoxicity. That's a big name for this process. So if this is an infected cell, here are these antibodies that are binding. Now remember, these aren't neutralizing antibodies. These are the other ones. They then bind to the cell, and then they direct parts actually of the innate immune system to come and then destroy that infected cell. So they're like scouts. And they say, hey, neutrophil, this is a cell, kill it. And so the point is that vaccines produce these guys as well. And so the vaccine, again, the people that make the vaccines, including PhDs, they are only focused on the neutralizing antibody, but there are other antibodies that are important as well. They sort of get shuffled to the side. Okay. What I wanted to say, this is one of the reasons I wanted to talk about T cells. Again, why some people may respond to a vaccine and some people may not respond to a vaccine. I don't want to spend too much time on this, but the point is that to respond to a vaccine, the T cells have to recognize that antigen, right? Now, in order for the T cells to recognize that antigen, you have to have the right MHC. So if this is the antigen and you have the wrong MHC molecule, you don't recognize that antigen and you're not going to respond very well to that vaccine. That's what this whole diagram is about. So some people just out of luck, they've got the wrong MHC molecules and they don't respond very well to a vaccine. So that's one reason that vaccines may work for most people, but not every person, because that unfortunate individual just doesn't have MHC molecules that are recognizing the antigens that the vaccine is composed of. All right, and so like natural infections, the vaccines will give you these memory cells. So vaccinations and protection is all about the production of these memory cells. And again, you have to think of the memory cells as giving you a head start when a vaccine, a pathogen, you're racing, from goal line to goal line. The vaccine puts you at the 50-yard line, and so you have time to respond to the antigen, to the pathogen before the pathogen overwhelms. Because if the pathogen wins, you're gone. Yeah.
Alan, so going back to the previous slide. When they're developing a vaccine, do most people have the same MHC molecule?
No, everybody has different MHC molecules.
So how do they develop a
But there's a
vaccine?
little slippage there. The linkage to the MHC is not as strong as the antibody. I mean, like I said, the biophysics of all of this stuff. So they broadly bind, and that's good enough. But some people, so there's a whole spectrum. And if you're way at the end of the spectrum, you're out of luck. Whereas most people, and that's why they test the vaccine. So if they have a vaccine that most people are not going to respond to, they will not put that forward. So that's part of the testing process of the vaccine. It's got to be safe and effective. That's the effective part. Is you've got to have if you're doing a subunit vaccine, that's what they're testing. Which subunit tends to work for the most people. And it only works for 50% of the people, then you got to start over again and try another.
So when they put the COVID vaccines out so quickly, they kept saying they didn't have time to test it. Was that what they were saying? That they really didn't know?
Well,
or was it is that a different technology?
well, they actually did have time. They did test it. It turned out that I don't know if they got lucky, but it worked pretty well. And what they didn't test, normally when they're doing that kind of the test was, it takes them, 10 years or something, but each test is a population of, 30,000 people at the most. And with the COVID vaccines, because it was an emergency, they gave, but turns out that it was right. They had a billion people that were immunized, and for most people it worked. When I showed you the slide, so in some sense it was a matter of luck. It could have been, but they did do some testing, but it wasn't as extensive. So essentially, the population was, when they do testing for these drugs, there are all different phases. And it's the last phase, phase 4, I guess, that takes the longest time. And so when you give a vaccine, how much do you give? So that too they have to test. And so in a sense, the population was the third or fourth level of testing. So they did do, it's not that they didn't do any testing, it's just that they didn't have time to do the normal vaccine rollout, which would have been a decade. And like I said, you saw the corpses piling up. Corpses are piling up, you don't have time to deal with that. You gotta do something quickly. And again, all right.
Well, you have a question that's been back there.
Oh, I'm sorry.
Yes, question I have is, some people say they have reactions like the aches or the chills, and after the vaccine is given, and other people don't. Does that mean that one person is getting more of an immune?
What was the last part?
Does that mean that someone that gets the chills and the aches and the side effects from the vaccine is more immune than someone who doesn't get the side effects?
Well, the answer to that is, I don't know the answer, the exact answer to that is that people are different. But the aches and the chills are essentially your immune system responding to that antigen. Now, if you don't have any response, you may not be responding to the vaccine. So if you think about it, whenever you get an infection, there's a period of time where you aches and chills. That more or less is your immune system kicking in. And that's what you feel when the immune system kicks in. So most people feel that for a day or two is when the immune system is actually being activated. That's the idea behind the vaccine. The real problem is what happens after that, for the real severe side effects, which it's already 11 o'clock, so I don't know whether people want to stop now and I can pick up later in 2 weeks, or you want me to keep going.
How much more do you have to go? Another 2 hours?
No, it won't be 2 hours, maybe, I won't go more than another half hour, put it that way. But I want to get into the side effects, which is the question that you bring out.
Can we go up
which
to
is-
the 40,000 foot level?
Excuse me?
Can we go up to the 40,000 foot level?
The side effects will be a little higher level. Okay.
Go for it.
All right, let's see, that's where we were now. So I could stop right now. So I had that time now pretty good. All right, the bad. Statistically, significant side effects of approved vaccines are rare, but when they do occur, they often involve inflammatory autoimmune disease. That's what the severe side effects really are. Now, autoimmune diseases are multifactorial. So, they involve the genome, they involve your genes, like I said, these HLA antigens and other genes, which I didn't get, and what we call the exposome. That's everything in the environment that might affect your immune system. And they interact, and you then have autoimmune disease. So, some of the environmental agents implicated in autoimmune disease / inflammatory disease, really same thing, can be viral or bacterial infections, vaccines, but then there's a whole slew of these other things here. Smoke particles, so all these fires that we've had, those smoke particles are linked to alterations in the immune system that can give rise to autoimmune disease. Microplastics, organic solvents, antibiotics, you name it. This is just a listing. So everything in the environment, and the point is they don't act one at a time. They act all together. So it's the sum total of everything that's taken place. So, one mechanism that's thought, and this isn't the only one, so when you're infected with say a virus or something, turns out that some of the shapes of the antigens, I'm going to go through this quickly, some of the shapes of the antigens, remember I told you all about shapes, may mimic some of the shapes on your cells. So that when you begin to respond to the antigen, well, remember I told you had these cells that are around that can respond to your cells? They get activated at the same time. They're not supposed to, but sometimes they do. So by this process of molecular mimicry, you can end up with autoimmune disease. And so actually the first example of that was with streptococcal infection. So I guess streptococcal infection is systemic strep infection is not as rare these days. It's not as rare these days because we have penicillin and other antibiotics. When I was growing up, not so much. What those strep infections did, people ended up with heart valve problems from scarlet fever. I don't know if anybody here remembers that. Well, what was going on is that the bacteria had a shape, molecular shape, that was similar to some molecular shapes on your heart valve. And so the immune response to that bacteria then targeted your heart valves. And so that was a side effect due to molecular mimicry of being infected by scarlet, by the bacteria. It's not just the streptococcal infection. All pathogens tend to have a potential to do that. So, COVID-19, we all hear about COVID-19, of course, and particularly long COVID. So what the hell is going on with that? Well, it's now clear that the spike protein has shapes on that protein that mimic shapes on neurons, on blood cells, on muscle cells. And so by stimulating a response to these shapes that are on the COVID virus, in some individuals, you stimulate these latent cells that recognize your tissue. In this case, Guillain-Barré syndrome, which is a destruction of peripheral nerves by antibody-mediated destruction of peripheral nerves, is stimulated, and this is a known COVID, long COVID side effect of being infected with the virus by this process of molecular mimicry. Now the point is that when you do that, when you get it, in order to mount an immune response, you produce these B cells that recognize those antigens, and T cells and B cells. The problem is, even if you get the virus eliminated, these cells still are there. You don't get rid of those cells. And so these cells still recognize these antigens. And it's self-renewing. And that's what long COVID, or one of the things that long COVID is all about, is you've stimulated, you got rid of the virus okay, but your immune system is now attacking cells that have shapes that are similar to that virus. That's what long COVID is all about. Now, CDC has recently recognized Guillain-Barré syndrome as a side effect of COVID vaccination. Well, it's not surprising. You're targeting the immune system to recognize pieces of that virus. And so what you end up doing is producing antibodies in some individuals, not everybody obviously, but a minority of individuals end up in trouble. But the point is, if they would have been infected by the virus, they would have been in trouble anyway. So everybody understand what I'm talking about?
So it wouldn't matter which vaccine they got?
In my view, in my view, it wouldn't matter. And in fact, it could be worse. And I'll show you the next slide. So, Guillain-Barré syndrome is not just COVID. It's recently been recognized for influenza vaccine. Now, this is what's really interesting here, that I found, is with the vaccine, it's 1 out of 1 million. So if you're getting an influenza, you have to take these numbers with a bit of salt. I wouldn't put my life on 1 in a million. And there are reasons for that, which I won't get into right now. But this is a CDC estimate, I think. So the estimate is if you get a influenza vaccine, you've got a 1 in a million chance of ending up with this neuromuscular issue. However, look at this. Just getting infected with influenza gives you a 17 out of 1 million chance. 17 times the... So the point is that actually getting vaccinated actually reduces your risk of getting this. And that probably has to do with the dose. Because if you're vaccinated, you get a little dose of the antigen. But if you get infected, you got a whopping dose. So it's more likely you're going to have problems. Again, this is one of the side effects of vaccines. Now, so the big question is, how is it that only such a small percentage of the population is susceptible to loss of tolerance and developing autoimmune use after a specific vaccination or infection with specific disease? Because these numbers, while real, are small. Like 1 in a million is not very large. Even 1 in 17 million is not very large. That's part of the problem when you tell people, when people say, well, we don't have enough data. It's hard to get that data because these are really small numbers, really small percentages. Nevertheless, they may be small, but they're real, and if you're one of the unlucky ones, you've got the problem. But the question is not why we have, the question is, why don't we have much more of this, is the question. I'll skip that slide. Okay, so generally, understanding vaccine side effects is difficult and part of a much larger problem. This is the point I want to make. So, autoimmune disease, that's the problem. So most of these side effects are autoimmune or inflammatory diseases. That's what we're talking about. 8% of the US population suffers from chronic autoimmune disease. As you get older, you get more likely. 80% of them are female. So, all of these things interact. We got genetics, sex, dietary components, toxic chemicals, gut microbiome, infections, and of course, vaccines, because vaccines are like infections. So all of these things are interacting simultaneously. So question is, well, why doesn't everybody get cited? Well, they may get the same vaccine, but their genetics are a little different. I talked about MHC, for instance. Other genetic features are important. They may have different diets. Certainly, they may be exposed to different toxic chemicals. So it's perfectly possible that if you've been breathing smoke for 2 months and then you get a vaccine, you may be more susceptible to a side effect. I'm not saying that that's the case, but the point is that all of these factors, there are multiple interacting factors simultaneous that act to give autoimmune and chronic disease. And it's actually even more complicated than that, because I told you that there's this randomness to the immune system. That's called your immune repertoire, what kind of shapes that your immune system can recognize. That's random. So people have different there's a randomness that goes into that. And then again, there's also a history. Turns out it's not just the side effects, these factors, it's the history of your factors. So if as a child you were brought up in an area, a farm, that had high pesticides, for instance, that likely affects you for the rest of your life. And it's not just, so what I'm saying, it's not just these components at any one time, but it's actually the whole history of these components. So the point is, I'm trying to tell you how complicated this is. And the point of vaccine side effects is just part of a bigger problem. And I will end this with, I know there are engineers in this community, and this is to give, for those of you who are engineers, this is a mathematical
Matrix algebra.
description of how engineers might look at the problem. And again, it's the interaction of the genome with the exposome. And so all of these features are a set of responses to the immune response, and these are your set of environmental inputs. And this is kind of a matrix, linear, looks like linear algebra. Again, if you're not an engineer, you can forget about what I'm saying here. And so it looks like matrix algebra, a linear problem. But it turns out that actually each of these factors are also an explicit factor of time, which I mentioned in history, and they're also a factor of each environmental input, so that actually this isn't linear, this is a set of nonlinear equations. And this is a very large number, so this just shows you how complicated the problem really is. Now, it seems that the NIH has just recently got the message, but going forward, I got a question mark, because I don't know what's going to happen with what's going on now. And this was the first attempt by the institute to fund, to begin funding of autoimmune disease as a separate entity. So right now, the description of autoimmune diseases, well, you've got people looking at vaccine autoimmune disease. You've got people looking in the environment, say heavy metals in autoimmune disease. You've got people looking at neurological autoimmune disease, multiple sclerosis. We're in neurology, so the neurological institutes. Actually, what they were proposing at this time was to create a new institute like the NCI, but an institute for autoimmune disease, National Institute of Autoimmune Disease, to put everything under one umbrella. And I can discuss this more in person if you want, but this is where we are now. And so when you hear people like, well, put my own opinion in, you've got Kennedy now as Secretary of Health, and he says, well, we don't have to deal anymore with infectious diseases, I just want to focus on environment and autoimmune disease. Well, what he doesn't understand is they're all connected. You can't get very far with studying one. You have to study them all together in a unified way. And so I'm going to end the talk in 2 hours and 20 minutes. Anybody have any questions?